Today we have Dr. Sachini Amarasekara presenting actually a study that she's also part of on the postpartum depression. Over to you, Sachini.
So good afternoon, everyone. Today's Science Club presentation is going to be a little different from what we had so far, because previously we talked about very common topics, day-to-day topics like COVID-19 and then some environmental issues, which was matching with the broad range of audience. But today, as you can see from the topic itself, sometimes you may feel like that this is little biased towards the IIME stream. So after giving a little thought, then I thought I will continue with this, because I think I can give an important social message also from this. And then again, because this is one of the projects that I was involved in during my PhD period, so I hope that you will gain some new insight into behavioral research, and especially the students. I hope you will see a lot of different types of data in here, and you will also study a bit to identify and interpret different type of data. So this was published in PLOS Genetics in 2019 by the DAC51 research group of the Laboratory of Immunology and Medical Microbiology, Department of Microbiology, Chungnam National University, South Korea. So as you know, a birth of a baby can trigger a mixture of feelings like excitement, from excitement to joy, then anxiety, or sometimes fear. So most of the new mothers, nearly 80% of them will have these mild depression symptoms, which is commonly called as baby blues. So worry, sadness, mood swings, and tiredness. Sometimes in some people there will be cry spells, and in 10 to 15% will have a little more intense condition called as postpartum depression, where you will have depressed mood or severe mood swings, difficulty in bonding with the child, and thoughts of harming herself or the baby. And there's a more intense case where postpartum psychosis. In postpartum psychosis, the patients will have confusions and disorientation. There will be obsessive thoughts about the baby, hallucinations and delusions, and attempts to harm herself and the baby. And when we consider postpartum depression and postpartum psychosis, these are clinically significant conditions which needs medical attention, and it can last longer. And the interesting thing is, not only women, even men can undergo postpartum depression, especially young fathers who have financial problems, and because of the changes in sleeping patterns and eating patterns during this period, they can also undergo postpartum depression. And when we talk about the risk factors, physiological risk factors of postpartum depression is well defined. So if you have a personal history of psychiatric illness, there's a risk of getting postpartum depression. And then poor partner, social, financial support, alcohol, drug abuse can also contribute to postpartum depression. But especially when we consider this poor partner and social support, support in the sense it can be emotional support like concern, comfort, encouragement, and then you have instrumental support, time, money, and other kind of tangible things. And then you have informational support like proper advice, education, and knowledge. So if a young mother that is coming from a low-income family is lacking support from the partner or from the family, then she has a high risk of getting postpartum depression. And especially in developing countries like ours, as I go through a lot of social media sites where the women especially in women groups, where they complain about their problems and so on, I can see that these problems with the mother-in-laws and the influence of elderly women who are too much concerned about their own son, or too much concerned about their grandchildren, have caused postpartum depression in lot of younger women and people. These people, they don't understand that these small sayings can be powerful enough to cause such diseases in young mothers, because from all what they go through, from all the hormonal changes and from all the heavy work that they have to do, so they become very prone to PPD. And it is very important to understand that even a small word, small comment can cause depression condition in these young mothers. And then we have many obstetric factors such as unplanned pregnancies and pregnancy complications and delivery modes. And finally, the genetic risk factors, where we are talking about today. And actually, the genetic risk factors are not well defined in case of postpartum depression.
So recently, they have found several genetic polymorphisms, like the serotonin transporter polymorphisms and catechol methanotransferase genes and BDNF gene as risk factors for postpartum depression. Okay, so in this study, we have found the deficiency of T cell death associated gene 51 as a risk factor for postpartum depression. And let me give you a brief introduction about this gene. So this was basically found in a T cell hybridoma cell line, and it has actually multiple. It has a function. It has functional diversity because, as you can see from here, it is involved in cancer. In some cancers, it plays a proto-oncogenic role. In some cancers, it plays a tumor suppressor role, and it has a play. It is playing a role in inflammation, then adipogenesis, apoptosis, behavior, vascular calcification, muscle regeneration. And I also couldn't include stress responses here. So this gene is also involved in stress responses. So to give you the background to this study, okay, before that, for the people with non-genetic background, now we have the wild type mice in here and knockout mice in here. So simply speaking, the knockout mice are the ones in which a gene is inactivated. So in here, the TDAC51 gene is inactivated. Hope it is clear for you. So if I give you the background, what happened is, now my professor has developed this mice, and when he tried to amplify them, his wife, who is a biology teacher, she was also involved in this work, and she has observed that all the babies that were born to these knockout females were dying by postpartum day 1. So this has continued for a while. They have observed this death several times, and then the professor asked the students to design an experiment, and we started this study in order to see what is actually happening in these female mice. Okay, so first what we did was we looked at their survival. So we took the pregnant female mice of the wild type origin, hetero origin, and knockout origin, and we when we checked it, we actually fixed cameras to observe their behaviors and observe their maternal behavior and all sort of behavior. So we could see that in the wild type cage and the hetero cage, the babies were nicely fostered, and the babies were gathered in one corner of the cage as in form of a nest. But in the knockout cage, the dead bodies were all over the cage, and there were even some parts of the bodies, like probably due to the cannibalism. So, but there was no difference in the number of pups that were born to all these three types of mice. And but the survival rate significantly declined by postpartum day 1 and postpartum day 2 in the knockout female mice. Then we wanted to check whether there are some developmental abnormalities. Actually, after generation of these mice, we checked about other parts, whether there's an abnormality in the spleens and all internal organs. We even checked the abnormalities in the bone marrow counts and osteocytes and all that, but we could not check these mammary glands. So in here, we first checked the mammary gland formation. So we could see that they had proper nipples. And when we checked in the, as you can see in the stained picture, the alveolar size was also okay. And then when we checked the suckling ability of the babies, we could see sufficient amount of milk in their stomach, so that we could decide that they are having the ability to suckle. So there was no problem with suckling, milk suckling. Then we also checked this cross-fostering, did the cross-fostering experiment to see whether the problem is within the babies. So when we changed the babies that were born to these knockout females to a wild type surrogate mom, we could see that they survived. But when the wild type babies were transferred to the knockout female cage, they did not survive. So from the cross-fostering experiment, we could see that the survival rate is still going down in the cage where the knockout female was there. And again, to see whether the genotype of the male partner can influence, we also added a wild type male and a knockout male to these cages and checked separately whether they can influence the survival rate of the pups. But irrespective of the genotype of the male partner, the knockout females still reported lower survival rate in their pups. So this continuously proved that there's something really going wrong in these female mice. Then you know that these animals, the rodents and especially mice and rats, they have this innate ability to make nests when you give them some cotton or some this kind of material. So even in the lab, they show this kind of behavior. But if they are not showing this kind of behavior, then that could be probably because of some of their behavioral problems or some health problems. So we checked this nest formation ability. We gave them some cotton material at prenatal day 3 and checked for the nest forming ability in these wild type and the knockout mice. And we could see that the wild type females, they make the nest very nicely, and it continues up to postnatal day 2. As you can see, the babies are gathered inside the nest. But in case of the wild type female, the material itself is scattered. And by P0, you can see the babies somewhat gathered into this area. But again, there are some babies scattered in these areas too. By P1 and P2, you can't see any pups in here. So that means they are dead. So when we gave a score for this nest, we could, so we gave a score from 0 to 5. and the nesting scores were also significantly reduced in the wild knockout female mice, and the gathering of pups in the next in B0, you can see only few are gathered in here. So gathering of total pups has also reduced significantly, further showing that they have some maternal care defects. And another important thing that these rodents or mice are showing, they usually grab their babies from the back of the neck, and if you move the pups into another region out of the necks, they go and bring them back into the cage. So this is called as pup retrieval. So when we move the pups, the knockout females, they are not that interested in bringing these pups back, but the wild type mothers, they quickly go and bring their pups into the cage, into the nest, sorry. And so when we check the percentage of pups retrieved, we could see again the knockout females have significantly low pup retrieval, and the latency to retrieve each pup is also increased up to 60 seconds, whereas in the wild type group, they do it within 20 to 25 seconds. And also when we looked at the nursing type of the pups, we could see that the wild type mice is nursing them for more than 300 seconds, whereas this knockout female is either not feeding them or they feed for very short period of time. Then after observing all these abnormal maternal behaviors, then we thought we should check for any kind of depressive-like behaviors and anxiety-like behaviors. So for that, we use some standard tests like the sucrose adaptation test, sucrose preference test, tail suspension test. So first, let me start from here. So first, we did the sucrose preference test. We allowed them to get adapted to sucrose for 24 hours using 1% sucrose, and then perform the sucrose preference test. Usually, when they are stressed, they don't like intaking sucrose. So as you can see, the knockout females, their sucrose preference has reduced in here. And then when we perform the tail suspension test, that is also for to check the depressive like behaviors. Usually when you hang the mice from the tail, they keep on moving, but when they are stressed, they don't move. So the immobility time is increasing. So in here, as expected, we can see the immobility time has increased in the knockout female, and the mobility time has decreased. Then we also perform the forced swimming test, again to test for depression-like behaviors. In here, when we add this mice into a water-containing jar, they tend to swim usually, and they just keep their head up and start swimming. But when they are really stressed, they just keep the head up and stay still, just like they look so hopeless, as in case of depression. So the immobility time, if it increases, that means they are in depression or they are under stress, and swimming time decreases in case of depression again. So you can see that the knockout female mice has high immobility time and low swimming time. Then we also check the anxiety-like behavior through this elevated plus maze test, where you have this cross-like structure with 2 open arms and 2 closed arms. When you keep the mice in here, they usually roam in these areas, but when they are really stressed, they are kind of close. They are fear of entering the open arm, so they tend to move more towards the closed arm. So we calculate the time they spend on the open arm and the number of entries to the open arm, and you can see that the time they spend on the open arms has decreased significantly in the female knockout females, and the number of entries have also decreased in the knockout females. So from that we could conclude that these mice are having depressive-like behaviors and depression-like behaviors and anxiety-like behaviors. Then now, if we have a depression-like symptoms or depressive behavior disorders, there could be, the reason could be some abnormalities in brain tissues or morphological abnormalities or functional abnormalities. So for that, we looked at their brain, a wild type at the knockout brain, and stained with the hematoxylin, and as you can see from here, there's no morphological difference in these brain sections. So we decided that there's no morphological changes in the brain. And when we check the expression of TDAC51 in the wild type non-pregnant mice brain, the neocortex and the hippocampus had significantly increased levels of TDAC51. And when we check the expression of TDAC51 from prenatal day 2 to postpartum day 2, again, from the postpartum day 0, the TDAC levels got increased. Then we wanted to see exactly where it is getting expressed. So we further conducted in situ hybridization technique. Okay, so so we used RNA probes in here, and we could see that the TDAC expression was increased in the neocortex, hippocampus, and the hypothalamus regions. And then we wanted to see in which type of tissues this TDAC51 is expressed exactly. So for that, immunofluorescence technique was used, and we used antibodies against TDAC51, which is tagged with this PE conjugate. Then antibodies against neuron cells tagged with AF405 conjugate, and anti-GFAP, that is again antiglial fibrillary acidic proteins conjugate AF488 conjugate. So the TDAC51 will be stained in red, and the neuronal cells will be stained in blue in here, and the astrocytes and all these astrocyte cells of the glial origin will stain in green. So when we merge, we could see that the TDAC51 expression is basically seen in the neuron cells, not in these glial astrocytes of glial origin. And we could also see that in the non-pregnant mice, the TDAC51 levels was low, but when you come to the P0, the TDAC51 expression is increasing. Okay. So then we conducted, we have this transgenic mice, which has TDAC51 expression in the brain-specific expression in the brain-specific expression of the TDAC51 gene. So when we use these transgenic mice, so if sorry if, for the people who are not familiar with these transgenic mice, let me simplify it. So transgenic animals are the animals whose the genome has been modified. So it can be a knockout mice or it can be a knock-in mice. So in case of this, if I simplify, just think it as putting back the TDAG51 gene that was lost earlier. Okay, so if you think like this, it's fine, especially the students. So when you introduce that back, we could see that the fostering ability of the mice was improving back. So they had the TDAG transgenic line mice showed normal fostering of the babies, and similarly their nesting pose also improved again, and gathering of pups, and all those tests that we did for anxiety and maternal behavior, they were improved and they got restored. So I will not, because of time constraints, I will not go into all these again and again. So it's the same tests that have been done: nesting scores, then gathering of pups, pup retrieval, again latency to retrieve each pup, nursing of pups, and sucrose preference, and other behavioral tests and anxiety tests. So, and then we also made another, usually when you do these transgenic experiments, we prepare 2 or 3 transgenic lines. So we also repeated this in the transgenic line 3 as well. And I did not include all those information to these slides. So we could see the same effect in the TJAC transgenic line 3 also. And then now as we know, there's a problem in maternal behavior, there's a problem in they have depression like symptoms, they have anxiety like symptoms. Then we wanted to check whether it is because of any neuroendocrine dysregulation. So first we checked. Now oxytocin is a neuropeptide hormone that is produced by the hypothalamus and secreted by the pituitary. So they help us to promote lactation. Especially it is important for milk ejection, and it can also help prolactin to stimulate milk production, and it is important for social recognition, stress regulation, and so on. So we could see that oxytocin levels of the brain have expression in the brain has significantly gone down in the knockout females, and even the oxytocin receptor levels have also gone down in the knockout females. And this is quite similar to what is reported in postpartum depression women, because oxytocin levels are reported to be low in human patients as well. Okay, so then we also check the serum oxytocin levels, and serum oxytocin levels also significantly decreased in the knockout female mice. Then another important hormone is estrogen, and estrogen also can promote maternal behavior. But in this study, we did not observe any change in estrogen or estrogen receptor expression. Then we also checked another set of neuropeptide hormones, which are important for the maternal behavior, like arginine, vasopressin, that is important again for maternal behavior and maternal aggression, brain-derived neurotrophic factors. So I think I should not go in detail about all this. However, what you have to remember is that these are important factors for maternal behavior. So in case of AVP, the levels of AVP again went down in the knockout female mice, but their receptor did not go down. And again, brain-derived neurotrophic factor, there was a significant decrease, but the receptor did not have any change, and PDYN also similar to AVP and BDNF, showed a significant decrease in this marker, decrease in the knockout female. And then again in NPY and PENK also, the levels of NPY and PNK decrease both in the receptor and NPY and PNK and PNK receptor as well. But and we were expecting prolactin, which is important for the milk production of these animals. We thought that it will also go down. However, the levels of prolactin significantly increased in the knockout group when we compare to the wild type and the transgenic groups, and there was no difference in the receptors. Similarly, corticotrophin releasing hormone also increased in the knockout females when compared to the the receptor got increased compared to the wild type and the transgenic counterparts. Then we also wanted to see whether there are some alterations in gene expression in brain tissues. So we carried out these microarray analysis. So in there, totally, I think there were 2,000 odd genes upregulated and 1,000, somewhere around 1,000 genes downregulated. But out of all that, we selected 7 pathways, which is closer to what we study, like maternal behavior. So that comes under maternal behavior. So we consider next in building behavior related genes, aggressive behavior related genes, maternal nurturing related genes, anxiety related genes, again maternal behavior related genes, anxiety disorder related genes, and depressive disorder related genes. Likewise, when we consider these 7 types of pathways, we could see that 39 genes were downregulated in the TDAG knockout female, and 31 related genes were upregulated in the TDAG knockout female. So there was altered gene expression in the brain tissues of these TDAG knockout female mice. And this was further confirmed using real-time PCR. So all that we found in here, all these genes that we found in here, we further confirmed with real-time PCR analysis. So in these, so I will not go in detail about these. So both these slides shows the confirmational evidence for the altered gene expression. So if I am to conclude this study, the TDAG51 deficiency in the brain caused some changes in the expression levels of maternal behavior and anxiety related genes, and it caused dysregulation of the neuroendocrine factors and monoamine neurotransmitters, which also comes under neuroendocrine factors. And because of that, it has caused abnormal maternal behavior. So we concluded that TDAG51 can act as a maternal care associated gene that may be involved in the development of abnormal maternal behavior and postpartum depression. So whatever the symptoms that we saw in these mice were quite similar to postpartum depression and postpartum psychosis. They were more intense than baby blues, common baby blues symptoms. So even when we take postpartum depression and postpartum psychosis, I think they were more close to postpartum depression. So that's why we focused more on postpartum depression in here, especially during this session. So the take-home message I have is, so you may have a lot of people suffering from postpartum depression, or one day if you are feeling depressed, especially this is for the students. So one day you all will become mothers and fathers, and one day if you feel sick after baby birth, don't be reluctant or feel embarrassed to go to a doctor and take proper medical attention. And then again, if you have mild depression, they will give you some counseling and mild antidepressant. Sometimes even during pregnancy, you can have these depression-like symptoms, so that again you can go to a doctor. They will recommend you some mild antidepressant. So the important thing is that you should not be reluctant, and especially the people who have these kind of problems, they don't admit it, and they don't acknowledge the fact that they have it. So they just try to be at home with the disease. So if you and the others, if you have any for especially for the boys, if your wife or one day if your friends are having these kind of problems, please, if you suspect that they have it, please help them, talk to them and help them to seek medical attention, and don't think that these will improve without medical help. So that is what I want to say today. And I would like to thank my TDAC 51 research group. This is Professor Jerang Ro, Dr. Jion Yu, Dr. Yoon Hyun Sok, the first author of this paper, and Dr. Kim Sumi, Nari Lee, and Songray Lee. So these were the people who were included in the TDAC 51 research mainly. So thank you very much for listening. And if you have any questions, you can ask now.
Sachini, to start off, I think it's one of the most interesting things. I think I have never thought about this particular aspect of genes in behavior too much. Sachini, are there any studies with the human counterparts of these genes in terms of postpartum?
No, sir, no. That's our concern too, but it's sort of hard to get human samples. I think the professor is thinking about that as well. But regarding TDAC 51, so far, no studies.
So you have a similar kind of a pathway in humans also, is it?
Yes, because the FILDA one in human is the TDAC 51 in mice.
All right. So you mentioned a knock-in study.
Yes, sir.
I think for the interest.
At the last part, the transgenic knock-in, yes.
Yes. For the interest, can you kind of elaborate a little bit on that one?
Okay, sir. So this is the one, sir. So in here we expressed TDAC 51 in the brain with the brain specific promoter.
So what I'm interested in is to know how exactly that was done.
Actually, you mean the procedure for transgenic generation, sir?
Transgenic generation in that particular tissue.
So when you use the promoter for the brain, the expression will be in that particular tissue, no, sir? So usually when
yeah, this is not like a selective knock-in like what we do for other organisms, like a conditional knock-in, not something like that, right? So this is like from the start you have the expression restored.
Yes.
All right.
Sachini, it's the 1 minute here.
Yes.
Sachini, thank you very much. It's a very interesting one. Like I even had never thought about it. I just wondering whether there is like genetically is it coming? Like I'm not having that genetical background, so I'm just wondering whether it's coming from mother to daughter, or genetically is it?
genetically in the sense, in the no, when you have now this is in knockout mice. Now we have this problem. So
Yes.
obviously, when the babies, if a baby is a knockout baby, then obviously that baby will also have this problem. So,
Okay.
but you asking about in human patients? Human patients, we don't have any data.
Okay. Yeah. Okay.
But so like now when we are amplifying these mice also, the problem is you can't do knockout breeding, because then all the babies will be dead. So
Yeah.
then we have to cross them with heterozygous females, and then do genotyping to identify the knockout females.
Okay. Yeah. Thank you, Sachini.
Sachini, thank you very much for a wonderful one. Talk here, just I wanted to ask, does this pathway have also connections towards the depression during pregnancy?
Depression during pregnancy, actually, because now in prenatal time also, when they are close to giving birth, yes, they show some symptoms, but not really in the first stages. But actually, to check on that, we have to check that further. But we did not have a look on the first period of pregnancy. Actually, in the prenatal period, as you can see from those nest building and stuff, there is a small issue. But our focus was mainly on the postpartum period, because that is what we observed first in from the professor's wife. She only observed this, and then we thought of focusing more on the postpartum period.
Thank you, Sachini.
Thank you, Sachini, for that very interesting presentation. Just curious to know, like you said, that the oxytocin levels varied, and it was seen in human as well. I'm just curious to know, as you said, like the prolactin levels was kind of increased in a way that you even didn't expect. Is that the same in humans? Like, has it been studied in that way, or
Prolactin, you mean?
prolactin? Yeah.
No, we didn't get any evidence, but there's a gap of information there, because actually there's a problem in understanding how this prolactin is behaving in this case. So even in the discussion now, even during revisions, we had a lot of trouble, and somehow we couldn't find the way that this prolactin is getting regulated like this.
Mm-hmm.
In human patients also, no information so far. But in human patients, sometimes when they have postpartum depression, they have low, is that what you're asking? They have low prolactin levels, and then low milk production.
Right. No, but in these mice, the knockout mice, the levels increased, right?
Yes, that's the thing. So that was very interesting, but we couldn't find how it is happening. So actually, further studies are needed on that.
Right. So that's kind of contradictory to what happens.
Yes.
Okay. Thank you.
I have a general question. Sachin, it's a beautiful study, not on the study.
Okay.
But if you know the general background to postpartum depression.
Yes, madam.
Is it now in one place you had said that, I think in the paper I read somewhere, that most stress-prone females
yes
can get postpartum depression. Is that it?
Yes, madam.
So then this gene is directly related to stress.
Yes, madam. As I told, I couldn't actually, when I presented the functional differences of TDAC51, I couldn't add the stress responses in here. This gene is directly related to stress response, especially oxidative stress and so on. We have our own paper from the lab. Apoptosis, oxidative stress and all that, this gene is involved.
So it's not because of that, but just because this observation that you went in this direction. Is that
yes, madam? Yes,
Okay.
yes. That this is mainly based on the observation, madam. Actually, these mouse were brought in for apoptosis and stress release related studies. But because of this observation, only we started this.
Okay, so then what about neurotransmission and this gene down regulation?
Yes, madam. So the only thing we could find was that there are some regulatory. So this gene is regulating some genes that are related to these neuroendocrine factors and neurotransmitters. But we didn't go further. What we assume is, madam, now this TDAC51 gene is having. If I look at the regions of this gene, they have this PQ and PH like domains which are like which can act as transcription factors. So we assume that this TDAC51 is acting as a transcription factor. So that is how we think that it is regulating this neuroendocrine systems, madam.
Right, then.
But that is just an assumption, madam. We have to check on that further.
Yeah. Now I was just thinking now we are all stress prone, and especially the younger generation.
Yes, madam.
Right? And whether the numbers of postpartum depression is increasing now. You have no idea, no? Whether the percentages
no,
are
madam.
increasing or no? Right.
No, madam. About the numbers, I don't have any idea.
Yeah, so this is a very good research area, isn't it?
Yes, madam.
Some things, open-ended questions.
Yes.
And where I suppose your professor is an immunology professor or a molecular biology?
He is an immunologist, madam.
Right. So I mean, together with psychiatry.
Yes.
I mean, like a group, a lot of good work can be done. Okay, thank you, Sachini.
Yes,
sorry, that was a very long question from me. I'm sorry.
No problem, madam.
Okay, right. Thank you.
So, madam, regarding the methodology of the rat study,
yes.
You have put a photograph of a rat cage.
Yeah. In here?
Yeah.
Okay.
Was it what was the exact method you have used to quantify the image data? Was it like a time-lapse imaging or some observational?
So it's just the number of pups that it's the number of pups that were born that was calculated in the first graph, and then the survival rate of the pups. So both were calculated, both were just counted.
Okay. And these maternal care and their caring on pups and feeding behaviors, was it observed manually or using some other?
It was through cameras. We have fixed cameras to each cage to look at the maternal behavior. But in case of these, I mean, the pup retrieval and so on, also pup retrieval next building, we did through cameras, but tail suspension and everything manually.
Okay.
I think what Praneti is asking is whether you have used any image, real-time image analysis work,
yeah,
right?
Something like that, yes. Like to measure the distance between the pups. So like for an example, to quantify the maternal care or the interaction with the pup, was it considered the
you
distance
mean in
between
here, the nexting area and so on?
Okay.
Yes, nexting area and so on. We have used a software. I can't remember.
Image.
Most of the time, but for these, it's only the numbers.
Oh, yeah. This is the number of pups. Yeah.
Yes.
Thank you.
Okay.
Just a suggestion, Sachini. I don't know how much it could possible, but these kind of studies should go to the general public, like the broader idea and the things like, because we have a lot of issues with the postpartum depression in Sri Lanka, like and we are lacking the scientific background to support it. Like if we like these kind of things, I don't know whether it's possible or not for in your side, but it would be great if it's in in a very simple language, like a small, like communicate with the general public, it would be a great thing to do.
Yes, I get your point. I will think of
Devangini,
that.
you came up with the statement saying there is a lot of postpartum depression in Sri Lanka.
Yes, madam.
Is that a validated thing you are saying, or is it just something that is
No, madam. I mean, like when we talk to, like, sometimes even we don't know that we have that. I think when we are talking with the friends and the things that we can see in the newspapers and the social media, even though it's not highlighted it as a postpartum depression, like there are many cases in newspapers, like with the parents do such things to kids, and like those kind of suicidal cases. That's
I think even it's not well reported.
Yeah, like it's not goes to the account into the postpartum depression. That's what I was thinking. Like,
yes, and people are not aware of it also.
So when the child is taken to clinic, I'm not sure, I'm very old, so I can't even remember how it was. Like when the child is taken to the clinic, or when I mean, there are regular visits, no?
Yeah.
The mother checked on now.
Well, if I'm being truthful, I haven't taken my daughter to the normal clinics here, but normally we take to the child to the pediatrician for their checkups in 1 month, 2 months. At the first time, 2 weeks, the doctor asked a lot of questions. Afterwards, they never asked about the status of mom. But yeah, in Australia, for my first daughter, I can remember there always was a checklist, and they asked to fill it and talk. But in Sri Lanka, I never. Yeah. I don't know whether the midwives do that.
Yeah, Dr. Devangini.
I haven't registered for that, so I don't have any idea whether they are coming and checking with.
Yeah, Madam, can I add
Yes,
something to that? Since my since my doctor, he preferred me to go for the public, for the government clinic. He always wanted to have that card, even though we apply for go for a private consultancy. So I attend to those public clinics. So they after the pregnancy, after few weeks, they asked us to come and visit. So in there, they fill a form to check whether we have sort of this kind of depressions. The government, they are doing it now.
Madam, I think everything depends on the place and everything, because in some social media sites, mothers have even complained about the clinics, like because of the comments that they receive in the clinic itself has make them stressed, madam. So I think it depends on the place, doctors and the nurses. So we don't know actually.
No, this is an area that can be, I mean, like, care can be given, right? Maternal care. Yeah.
Yes, madam.
Because everything else is good. I mean, we can't complain. I mean, I know we are very great at complaining and putting down our country, but I think generally speaking, things are okay, isn't it? I mean, compared to our economic status and so on.
Yes, madam. When we talk about this support, we have the best informational support, I guess, madam. The only thing is that poverty, and then this myths and all these problems coming from elderly ladies and all that. So if we can get rid of those things, and especially if the husbands can be little more understanding, I think this problem can be solved up to a certain extent.
Okay, all the young men, please note. Right. So any more questions?
Yeah, the figures show that it's about 30%, the prevalence. That's quite high.
That's quite high, isn't it? Yeah. This is in Sri Lanka?
Yeah, that's in Sri Lanka, yes.
Wow. Okay. Right. So it's an interesting area for a multidisciplinary study to be initiated in Sri Lanka.
Exactly.
Yeah.
Yes.
Yes. So shall we wrap up if there aren't any more questions?
Yes, madam.
Right. So once again, thank you very much, Dr. Sachini, for that very thought-provoking presentation. And thank you all for the questions. I mean, we had a good discussion. So thank you and have a good day, everybody.